And with all that said, I think I will turn my camera off and the stage is all yours.
Thank you, Tiago. I'll just share my screen first here, just a second. Okay. I hope you can see it okay.
Yes, all good.
Okay, good. Just make some room here. Great. Okay, good. So first of all, thank you very much, Tiago, for the invitation. So Dr. Tiago and I, we are old friends in the scientific environment and also the privatosphere. So we have met in Berlin many years ago, and we kept this nice friendship and collaboration. And I'm really glad that since when he moved to Japan, to Tokyo, he is being very successful in his activities towards bioinformatics, computational biology, and data science. So, and I really welcome it, and I'm very thankful that he invited me to give this seminar to his colleagues in Japan. So it's actually the very first time that I give a talk to a Japanese audience, and it's a pleasure. So I had the opportunity in the past to visit Tokyo, and I really liked, it was a very nice place to be. New experiences for me, as he said. I come from Brazil originally, and trust me, Brazil is very different from Japan in terms of what you see there, but it's a very excited place, very exciting place to actually go and visit and have a really nice experience. And we have a lot of Japanese people, descendants from Japanese people in Brazil. So I think you are very much aware of that. So it's like you may not think, but the culture of Japan is really very present in Brazil, and this brings us a little bit closer if you think about how both cultures find this kind of interface. A little bit, I think. But when I was in Tokyo, it was a really nice experience. Hi. Really nice experience, and I'm taking the opportunity now to make this a more professional on the scientific side now. So I'm going to give this talk about patient data analytics today, and this is going to be on the scope of the academia and the industry collaborations that I've been working in Europe in, let's say, the last decade. So it's been 10 years since I really started doing this type of work in a very intensive way in Europe. So I've been in Europe since 2006 now, but really intensively working with patient data analytics. It started something about 9-10 years ago. So currently I'm associated to the information technology for translational medicine company in Luxembourg, which is based on the House of Biohealth, as you can see up here in the picture. So this is part of the Luxembourg Health Tech Cluster, which is a conglomerate of different companies that they have to do with health informatics, health technology. And because you are a National Center for Childhood Health and Development, I think that it's quite interesting for you to know about the existence of this health tech cluster, which might open the doors for future collaborations between Luxembourg-based companies or universities and you. On the public sector, ITTM is actually a spin-off company from the University of Luxembourg, and I am one of the co-founders of the company. And at that time, in 2015, I was a member of the Luxembourg Center for Systems Biomedicine, which is a very close collaborator, and also we have directors that are shareholders of the company. It's a very tight and kind of close interface in between the public and the private sector in Luxembourg. So, and this is what I'm bringing a little bit to you today. So the projects that we did at the time, that was at the LCSB, and then how I moved to London, and we continued some of the concepts in London, and then came back to Luxembourg in this kind of a private sector initiative. So yeah, we can find more information about the company on the website that's down here. And also I have my private address, that's adriano-barbosa.eu. So you can have like an overview of all the projects that I've been working since 20 years, more or less. And of course, the ones in patient data analytics on the last 10 years. So this is like to set up the scene, like as Tiago said, I started my career at the European Molecular Biology Laboratory. So as a PhD intern, and I was basically, associated to the group of Pierre Bork and Reinhard Schneider there. So we developed some web services based on SOAP for multi-omics patient data retrieval. And then I moved to the Max de Bruck Center in Berlin, where I met Tiago there. So I was doing the data integration of a neurodegenerative diseases network project called NeuroNet. And the idea was to bring together multi-omics data based on RNA-seq, protein-protein interactions, cell phenotypes, and literature data. And then we deployed some databases and also text mining software for doing that analysis. So I moved back to Brazil as an associate professor at the Federal University of Paraná. And we did a lot of RNA-seq analysis of microbia there. We published some work on that. So there are some pipelines available out there. So if you're interested to run RNA-seq pipelines and you don't know where to start, that could be an interesting start point for you maybe. So then starting out the part I mentioned before at the Luxembourg Center for Systems Biomedicine. So there were two major projects that I will detail a little bit more for you. And this is it has to do with multi-omics patient data integration. So finally, I was a recipient of the Rutherford Fellowship of the Medical Research Council in the United Kingdom. So I moved to the UK at the Queen Mary University of London during 3 years. And I was doing the data integration between two major data sets. One was called the UK Biobank and the other the BARTS Bioresources. So they had a complementary information about subjects like patients belonging on these two studies. And then we just brought them together in an interface I will show. And then on the last years, it's not 6 months here, it's wrong. It's actually 12 months. I was at the Medical University of Vienna as a senior postdoc for artificial intelligence applied to medicine. And then the idea was really to make a kind of a data mining in all the methods of artificial intelligence, but published in the papers with code resource. And we wanted to extract those methods from different technologies and map them in a kind of a trajectory map so that we can have an idea about what are all the technologies available out there for artificial intelligence tasks and how they could be applied to a different field. So we needed to develop a map, and this is something that I will talk to you in the next seminar because the paper is not published yet, is in press. So I hope it's going to be published in the next 2 to 3 weeks, probably. Let's see. So as you could see, I've been basically working with a lot of data. So, and most of this data is patient data. So it's coming from hospitals, it's coming from research projects, it's coming from collections of databases or biobanks. And then the idea is to extract information from those sources and then organize data sets and allow people to discover insights, scientific insights within those data sets. And this is a new paradigm for data analysis, right? There are many different papers published out there that tells that the idea is that you don't look to one specific mechanism anymore, like your favorite gene or your favorite protein. You have to have a kind of a more holistic view of the biological system. And one way of doing this is approaching, for example, single cell data. You can extract data now, not from a tissue specifically, but specific cells and try to cluster, for example, the pattern of gene information, of gene expression across different cell types. And then, of course, you can compare disease states with normal states. Let's say you get a patient that's suffering of one neurodegenerative condition, and then you want to compare the data collected for that patient with a normal control or within the same patient across different, let's say, levels of the disease. So if you could track from the beginning of the disease diagnosis until a very late disease symptomatic phase, you can compare what were the changes in gene expression that happened in different cell types of that patient in the course of time. And this is something that could be possible to do, and not only in the gene expression level, but also when you think about the proteomics. So now, I mean, recently in 2014, we had like the publication of this draft map of the human proteomics, which allows us not only to look at the gene expression data, but also the protein expression. So the idea is like, which are the proteins that are important for specific, let's say, tissue neurogenesis, or let's say tissue degradation when you think about a disease that's occurring in the course of many, many different years. And then you want to see what are these patterns of change when you look like a spinal cord or lung, liver, etc. So you get the information about the whole proteomics data available at a multi-organ level. And then you compare these patterns of protein expression across, these different organs or disease states. So, and then this is quite interesting because then you can put together the data of the proteomics together with the gene expression and see if there is a modulation in between the stages of gene expression, the production of the so messenger RNA, and then the protein output. So everybody is familiar that you may have some levels of post-transcriptional control of gene expression that most of the times are executed by microRNAs. And then this is something that you can discover when you compare the gene expression against the protein output levels across different organ systems. So, and then, well, like there are many different applications that you can do this. Like it creates a completely new paradigm for data analysis. You can see that this can be applied to different research areas, including what we are having now. Like we have the great interest of understanding how the coronavirus interacts with the human host. So, and then if you can do this kind of protein interaction maps in between two organisms, of course, if you consider the virus like as an organism, then oh, sorry, I think I'm making some background conversation. I'll just ask some colleagues some colleagues should maybe mute their microphone because I think it's a question, but at the end it's just someone talking. Okay. Mute. Okay, good. So I just mute someone. And then, so then you have this possibility, right, to put in context what you see in terms of gene expression, protein and protein-protein interactions across host, let's say host and virus, right, coronavirus interactions. So of course, there is a lot of data and there is a lot of challenges when you think about all this data that's being created. There is a data deluge. So, I mean, how do we data scientists handle this amount of data that's coming to our hands? It's of course being categorized as a big data. If you think about the amount of data, the velocity of the generation of the data sets, and also how dynamic this is nowadays, right? We just saw now that last week, a new company in the US developed like a gene sequencing method that costs less than, like reaches the target of $100 in order to reach a human genome. Imagine, like you have the possibility now of sequencing the human genome for $100 only. Imagine the amount of data that, this kind of technology can generate. So this is the first challenge. The second one that I illustrate here in this slide is the reproducibility. So if you want to reproduce your analysis and make sure that other scientists, they can, redo your analytical pipeline, and they can also get the same results as you did, like in a posteriori approach. So you have to make sure that your information is reproducible and documented in a way that everybody can, let's say, reproduce that, right? Like repeat your analysis. And then the big thing, which is the health data privacy, right? In Europe, you have the General Data Protection Regulation, which is a set of rules that everybody working with data should be aware. And one of the most important things is that patient data should remain unidentifiable and should remain strictly secure on your database systems, because you have to make sure that the reason why the data was collected was the same reason why people analyzed it. You cannot just like collect data from people that did not consent for a more broad, let's say, type of analysis. You have to make sure you use the data for the reason why the patients they consented for. And this is something that can lead to very, very expensive fines here in Europe. So we have to make sure that we use the data in a very legal way, in a very responsible way. And that's why I illustrated GDPR here. So just continue here. Okay, another paradigm of change. So when I was in London at the at the Queen Mary University of London, I was also working as a consultant data scientist for the National Health System. And the NHS in England has this approach to transform traditional medicine into personalized medicine. The idea is that you don't treat the disease anymore, you treat the patient. And then every disease can be different in different patients. So the idea is that you have to characterize what are the groups of patients that exist out there, and then try to profile each individual patients to see what are the best treatments for this individual there. So, I mean, this approach that one pill, like one pill fits all, that, you think you can handle a disease like in the same way across different patients, it's just fading away. So you have now really to look at individual patients and their molecular features, their laboratory features, and make sure that the treatments available out there is going to be the best one for the patient. And then the best way to do this is actually to collect multi-omics information from those patients. So you have like all the technology available out there to access like cell types, chromosome information, DNA, RNA, protein, and metabolic biomarkers. You can get these biomarkers and put this information together as a phenome information of the patient. And then you can try to see how the treatments available out there alter this phenome, and then see whether or not those treatments, they are effective in handling the conditions that the patients display. And cancer is one of the things that is right on the spotlight of this type of research. So you have first to characterize all the different types of tumors, let's say, that are identified in a set of patients. And then you can track what are the best treatments that work for specific tumors. And then if there is a new patient that resembles to an old one in which a nice treatment worked out properly, then you can suggest this treatment for the new incoming patient. It's everything about, getting the retrospective information of the medical practice. And then we know which treatments worked or not for which type of patient. And then now we compare this patient to a new one and then see if, this kind of a clustering, this kind of a similarities, they can be reflected also in the therapeutics. And this is something that works in a molecular way, but you have to make sure that it works also in the clinics. So the idea is that nowadays what we have is a very hard definition of what a disease is. And in the World Health Organization, you may know better than me, you have the International Classification of Diseases Code. So this is an international global accepted tool for diagnostic and use in epidemiology. And the idea is that you can manage better the health systems using like a coordinated naming of diseases. So it's a resource that's maintained by the World Health Organization. And the idea is that, you can semantically align the diseases across different countries. Imagine I'm coming from Brazil, I live in Europe, and you guys are there in Japan. And we want to make sure that we are naming this condition in the same way. But as I mentioned before, it might be that one named disease is not really what's happening across different patients that display something that is similar to that disease. We have to depict the molecular signatures and maybe try to reclassify those diseases using more information than just what the just what the doctor sees in the patient, or just what we have as a kind of a written in stones, let's put like that, right? Nomenclature. It's a dynamic resource, it's very useful, it's been out there since many years now. And the ICD-11 is the new classification code used in the world nowadays. There is a gap there. No, there is some years that they will adapt from the ICD-10 to the ICD-11. And then in the course of the years, we will bring more information to reclassify the taxonomy of diseases, which is something that might integrate not only what the doctor sees, but also what the scientist sees. So it's the idea of the translation of medicine now. You get the clinics features and you get the molecular networks, the laboratorial biomarkers, the genetic biomarkers, put them all together, and then you try to recreate the taxonomy of those diseases. And what allows you to do that is the availability of patient data, right? If you don't have patient data available out there, it is really hard for you to create a new cohort that will represent all the diseases available in the world, and then try to do this kind of a reclassification. So it's really like a way that you reuse the information, you create new modules to annotate one specific disease, and then now you can validate these modules of, let's say, sub-diseases in the clinics and see if it makes sense to bring new treatments to treat a specific disease, let's say, subclasses, right? And then this is what's going to make a new taxonomy a little bit more patient-oriented and also, let's say, modern and dynamic, because then you don't need to create a new disease classification. What you need to do is just subclassify what exists out there. So we have been doing this in Europe. So I worked in a project called Ethionomy, and the idea of this project was to reclassify neurodegenerative diseases based on molecular mechanisms. So this project was a 4 years endeavor between different companies that are listed up here, and also universities and research centers. You can see here University College of London, the University of Luxembourg. You can see the Erasmus Medical Center in the Netherlands, the University of Aix-Provence in Marseille, France, and then some pharma industries like that you might know. You have here UCB Pharma, etc., etc. So it was a very interesting project. Like, we were at the interface in between two major work packages. So there was a work package of data collection and the work package of knowledge management. And we were sitting here in the middle, so integrating with two different work packages. So I bring this slide also because maybe it's a nice way to illustrate to you how the consortia projects, they work in Europe. I don't know exactly how does it work in Japan, and then maybe I thought it could be interesting for you to know this illustration. So the work package 2 was a joint work of different institutions there. You have like a Fraunhofer Gesellschaft, the University of Luxembourg, where I was work, University College London, and so on and so forth. So the idea is that we had like specific people from the work package 3 collecting indices that could represent the disease in a better way. And we had a work package 5 that collected different types of information from different cohorts. You can see here Stockholm in Sweden, Barcelona in Spain, Paris in France, and Bonn in Germany. So we received this information that can be very, diverse. And then the work package 5 will fill those indexes informed by the work package 3 with information. So what we do is like we receive this, we promote a data curation, we store this information in a database, and then we integrate analytical tools from third-party groups into this kind of knowledge management interface. And then what we have now is the possibility to use the analytical results of those tools together with the stored data in our database to create new disease models. And this disease model will allow the users at the very beginning to do data analysis and to enable this kind of a new creation and brainstorming about how neurodegenerative diseases should be classified. So this is like a scope of a work package that I worked for the University of Luxembourg. And then maybe in the future, when we do, let's say, projects in cooperation, the National Center of Child Health and Development could be one of those partners here in the middle like providing us with data or with analytical tools that we could integrate in a pipeline for data analysis. So this is also like an extra collaboration that I did. So I did some text mining analysis there, but I will talk about this a little bit in the future. So in terms of data curation and management for this project that I just showed, we use like a specific tool called tranSMART. And tranSMART allows us to do this kind of, it's a Windows-like interface. Let's make some marketing here. So it's basically, it allows us to bring together different types of information in a very comprehensive way that users can just go there and navigate and pick up what's the type of information that they are interested. And then we plug this kind of, curated information into an analytic view. So we have like analytic tools that allows us to, let's say, mine the information that's deposited there and come up with like new insights about the diseases. So the idea is that this is one integrated interface, just called tranSMART, and then you have an attachment of analytical tools to data that is sitting in the warehouse. So we did some work on that. So you can see on those publications. So we created some standard way of organizing the information coming from different cohorts in a, let's say, master tree. And then this master tree allows the comprehensive navigation of different data sets in the tool. So we also created an integration layer between the data that's sitting on the database and a Galaxy interface. So Galaxy is a platform that's very popular in bioinformatics. So many algorithms and data analysis pipelines are implemented using Galaxy. And then, we thought that that could be interesting to create this layer that you just pick up automatically data from tranSMART, you plug into Galaxy, and then you export this to a disease map, for example. And then finally, we recreated the whole analytical interface of tranSMART using SmartR, which is an open source platform that allows you to do this kind of analysis in a very dynamic way. It's something like an update of graphical user interface for those users using our analytical tool. And I've been part of all those publications. I supervised people, I developed mine myself, I developed modules. So it comes like, on demand, it depends. And as I told you, this technology can provide you with like new powers to analyze patient data. So you can run hierarchical clustering, you can plot like an interactive box plots, you can create like different types of analysis, such as correlation analysis, or really there is a dedicated interface we created for the Ethionomy project called Brain Mesh, that you bring the data from the database and you plug into analytical displays interface that makes the life of the research easier. And also here, because we had also brain information in that project, we wanted to visualize how the volumetric and connectivity data about the brain could be explored in the interface. So all these kind of new technologies allowed us to actually win prizes in the course of the way. I went to the University of California in San Diego some years ago, and our work was featured as the best poster, the best work presented there for the new technologies towards tranSMART. And also, you may know here the Michael J. Fox Foundation for Parkinson's Research. So we also created, we won a prize there about the innovative technologies for biomarker discovery in Parkinson's disease. So that was something that motivated us to do our work further. So after this work, I moved from the University of Luxembourg to the Queen Mary University of London. As you see, where is it? As you see here, it's somewhere in London. Here we go. So Queen Mary University of London. So it was an initiative of the British government called UK Health Data Research Alliance. And the idea is that the UK Research and Innovation Agency was funding fellows all over the UK. And we had 5 different cohorts of scientists. And I was based in London together with other people in different institutions. And those were my fellows there. So you can see here the Charterhouse Square in central London. It's the place where John Vane worked in the past. And William Harvey also, in many years ago, almost in the Middle Ages, you know, he developed like the basis of the blood circulation in the body. And I was working there with different people, including people from the Genomics England Center. And it was quite interesting work. So what we did there, as I mentioned in the beginning of my presentation, was the integration of two major resources, the UK Biobank. So you have here the web interface of the UK Biobank with different, patient data from the British population. So it's a study. So patients that they receive medical care by the National Health System, they might choose to participate on this study. And the idea of the study was to determine what are the genes that have something to do with the pathology of rare diseases and cancer. And the idea is that if you have an external data set that you want to compare with the UK Biobank, you need this layer of semantic alignment. And what I did there was to create this very specific mapping tool that allows different data sets to be represented in the same way as the UK Biobank inside the tranSMART tool that we used for ethionomy in the past. And now we have like a more, let's say, integrative view of the UK Biobank data set. So it allowed us to tackle 11,870 clinical variables collected by the UK Biobank in this navigation tree here, as you can see. And now if you have like a new study, you can map your variables to those variables and then compare on the fly what are the similarities and difference across different patients groups, let's say the Tokyo population and the British population. So that would be a possibility by this project. So we also created like an inventory based on the ADNI study. ADNI stands for Alzheimer's Disease Neuroimaging Initiative. It's a project being developed in the United States. I mean, actually it's coordinated by the United States, but it has like patients that are, scattered all over the globe. And the idea is that, it's the same thing. I mean, you get the data from a major project and then you represent this in a more friendly way so that new data sets can be integrated and compared to our, let's say, reference project. And in this case, you can see that there is a lot of data out there, clinical assessments, biospecimens, genetic data, medical history, like family history of the patients, what we call subjects characteristics, and also information about the study. So everything is organized and integrated on tranSMART. So what you can do now is like once that the data is sitting on the tool, what you can do is like you can attach your analytical methods. For example, my team in London was interested to develop a machine learning pipeline. So you can see here this artificial intelligence pipeline written in R. So every time you see a little book like this here, so it's a leaf, it's one step of an R script that's running in the background. So if you're not familiar with R, you should, because it's computational statistics. So you have here different methods that are used in a pipeline. And then, these symbols here will represent like input and output of data from the database. And then all of these plots here are information that you can fetch back and plot on your interface. So what we did is like this machine learning interface at a database, and the outcome, sorry, wrong one. Yeah, I will have to show you later. So the outcome is like a different like analytical plots. I will show just in a second, right? Just to keep the course of the presentation. So that was the proposal. The proposal was to allow analytical methods to be attached to a database system and then run those analyses on the fly. In order to do this, you need to have access to research infrastructures. So the idea is that initially when you develop those systems, you're running with your local laptop or your local workstation in the lab. But because you want this to be available for a broad perspective, the idea is that we use the European research infrastructure, which is called here Life Science RI. So European Life Sciences Research Infrastructures. It's a cooperation of different research infrastructures providers in Europe. So, and then my colleagues and I, we wrote a little project to have access to those research infrastructures. And then we could do this kind of analytical interface that I showed you in an encapsulated way using the research infrastructures. So we don't need to have the computer power in our side, in our institute anymore. We just apply for access to those research infrastructures. And in this case, my project had to do with the identification of cardiovascular biomarkers for developing risk score of cardiac diseases. And what we did was, we used like different biobanks all over Europe in order to search for data of cardiac patients. And then in a second moment, we use a research infrastructure provided by, that was BBMRI ERIC. And then finally, we used EATRIS to liaise how my requests would be interpreted by the research infrastructure. So it was a kind of a successful project. The work was featured here as a success case story for the Corbell project. And this is how you could collaborate with us. So if you don't have the infrastructure on your lab, you could apply as a third-party set of researchers or independent researcher to use the research infrastructures of Europe. And, sometimes it's free of charge, sometimes you have to pay. It depends on the scope of your application and, of course, the availability of resources of the life science research infrastructure. So that's me and my colleagues. So these are all the people of the life sciences research infrastructure. A key person here is Nicholas Blomberg. So Nicholas is the director of the Elixia. Elixia is the European Life Sciences Research Infrastructure in the whole European context. So he's like the central point that you could have access to many different research infrastructures out there. And these are really cool people. You can talk to them and explain your projects. They will be really happy to help you with your endeavor. Yeah, so this is what I promised before, right? I thought that this slide was in the right order, but it's not. It was not, unfortunately. So this is like the outcome of the artificial intelligence algorithms that I showed before. You have here, I would just focus on the rock curves. So you can see here that the classification of different classes of patients, being controls, mild cognitive impairments, or dementia, they can be really maximized by using artificial intelligence. You have an overall here area under the curve of 0.90. So I would say that this is 90% of accuracy when you try to classify patients across different groups of diseases. So it's really powerful technology. So we also applied this kind of machine learning technology for clustering patients into different groups. So you can see here the possibility also, like the area under the curve is really high, 0.97, meaning that once you establish the clusters of patients, you can easily place new patients into those clusters by using machine learning. So the classification reports here in terms of precision shows that it's really powerful. So you have always over 0.85 of accuracy there, which is promising as a user. And you can also try to study what is the transition of patient groups in the course of a project. So here, for example, in the baseline readout, you have more patients that belong to the low depression and anxiety scores. And then you can see that there is not so many people suffering of the acute inflammation. So artificial intelligence methods can allow you to predict to which class, those patients will transit in a second follow-up visit. And you can see here that there is an increase of inflammation prediction. So this is everything like patient data derived. So if you have the right data hands and you use the right models, you can do this kind of trajectory predictions using machine learning. Yeah, just for you to have an idea how diverse this can be, right? You can see here that most of the patients from this class here, they have been divided in different other classes. And you want to know what is the possibility for transition if the treatment is working or not in a patient group. You can plot these nice Sankey plots, and it will give you an idea of the follow-up of the patient. So now what I'm going to talk to you a little bit is about our cancer project. So the cancer project used this kind of concepts in machine learning. And the idea was to discover what are the genes that could tell that a cancer is very aggressive in comparison to non-aggressive cancers. So we know that different pathways are involved in cancer, right? And, a pathway is composed by different proteins that work together. So one very classical pathway for cancer is the Wnt pathway. But there are several different other pathways that they are interconnected to this Wnt pathway. And it makes cancer to be difficult to predict, difficult to analyze, and difficult to understand once that you have different networks working together in the cancer phenotype. So it's very difficult to predict what are the genes that could be an indicative of an aggressiveness of a sample. So if a patient with this type of cancer will respond well or not to a treatment. So what we did is we accessed a cancer resource called the Cancer Genome Atlas, TCGA, and we downloaded information from 1, 2, 3, 4, 5, 6, 7, 8, 9 different types of cancers. And we plotted a correlation in between aggressiveness of the cancer and the level of entropy of the subnetworks of those genes. This means how diverse is the amount of connections that one gene displays in that network. So then we have highly aggressive cancer and low aggressive cancer. So you can see here the high levels of entropy and high levels of, let's say, survival, or low levels of entropy and high levels of survival. It depends. We don't know. It's a very difficult scenario there. So the idea is that we divided those cancers into 6. I mean, this is the classification, right? We are talking about 6 different cancers like stomach, lung, liver, kidney, thyroid, and prostate cancers. And we want to know what are the genes that will divide a highly aggressive cancer to those of a low aggressive cancer. So what we did was we got the genes of those networks I informed you before, and we accessed the levels of gene expression in those for those genes. Then we mapped what are the genes that are highly expressed with the levels of partners in an protein interaction network. So, and then we normalized the counts of interactions observed in a sample based on the expression levels and in the number of protein neighbors that one target gene is connected to. So, for example, so if you have a count of one specific gene, let's say the gene ABL1, that was upregulated one time in 125 samples that belongs to the class highly aggressive, we have a normalized count of connections of 0.008. And this is going to be the numerical representation of gene expression and protein-protein interactions values combined for that gene across the highly aggressive cancer type. So we do this for all the genes that are available in our pool of genes, and we try to reduce a little bit the complexity of these gene collections by using principal component analysis. So at the end of the day, what we have is 3 principal components that will capture something about 88% of the variance observed across the different cancer types. So with these 3 principal components, we were able to distinguish here the 3 less aggressive cancer types from the 3 highly aggressive cancer types. And from all the genes that we used, this is the top 10 list of the most representative genes that would give the signature of aggressiveness. Now that we know what are the genes that are the most relevant to distinguish these 2 classes of cancers, we were interested to know what are the genes for one specific class that could classify one sample as aggressive or not. So we learned, first of all, the differences between the classes. And now we want to know the genes that will make a signature for one specific sample to be aggressive or not. And that was the list that was most interesting for us. So this list allowed us to pinpoint the genes that would reduce this variability of, of a decision tree, like we use like the random forest classifier, to classify whether or not a sample is very aggressive or not. So if you work with cancer, you can see those genes, and there is the paper like mentioned there. Every gene has something to do with previous cancer mechanisms that have been reported in the literature. And what we did in the paper also was to put in the context of the literature, what are the genes that are most important for the classification of the different cancer types and for the classification of individual samples. So what we could see is that all those genes are somehow connected using literature text mining and also protein-protein interaction. So our idea now is like to make those genes available for a prospective clinical trial and try to see whether or not they are drugs that they interact specifically with those proteins from our, our list. And then maybe those proteins could be nice targets for drugability projects, which would reduce the aggressiveness of genes. So our hypothesis is that genes that are highly connected in the network and they are somehow highly expressed in aggressive tumors, they should be the best targets for a treatment, because then, that could be creating a kind of a bypass in the network and making the most aggressive cancers to be more resilient to treatments. So that's our contribution to the cancer community, is like a list of genes that could be really used in the future for cancer treatment. So I'm coming to the end of my presentation. So I just wanted to summarize what I discussed about, that diseases that should be considered on a personalized manner, not in a collective way. So patient data analytics will enable the discovery of new disease subtypes. So it's important that the data creation and integration are used out there so that you allow patient data to be analyzed across different cohorts. So it's a good idea to use machine learning algorithms for finding patient subgroups or select patients for, let's say, clinical trials in the future. I also showed the use case here of cancer aggressiveness that can be discovered and predicted using a data science, including machine learning approach. All these analyses that I showed you, they are used using reproducible notebooks. So it's really important to keep your code clean, readable, and reproducible using different platforms like GitHub, Jupyter Notebooks, etc. And of course, right, if you think about international collaboration, this is what really empowers new drugs discoveries that can work not only for one cohort, but for the whole world. So this is like some side projects of mine. So if you're interested in text mining, you can browse later on those papers. I've been working like really extensively on text mining. So it helped us to integrate the data science results into what the literature reports to us. So there is a lot of data analysis out there. So these are some international engagements from my side. So I'm very engaged with the German Ministry of Science and Research. So in the Bragforst initiative, we have like some seed funds between the Fiocruz Foundation in Brazil and the Queen Mary University of London. This is from where the grant for the cancer project was obtained, like via this collaboration. There was a lot of integration also in the international scenario regarding the pandemic when I was working at Queen Mary University. And yeah, as I said, like a university, the Fiocruz Foundation in Brazil, it's, I mean, the biggest epidemiology institute in Latin America. So it's a really good idea to make integrations and courses with those people. So I've been teaching bioinformatics and computational biology in different universities. So highlight here for a course in training on bioinformatics at Cambridge University. So there was like an autumn school there. I've been teaching different students from medicine, nursery, like health professionals. So the idea is that they have no background in machine learning, and I provide this bioinformatics training to them so that they can implement their projects from the beginning to the end in an autonomous way, and of course where to pick up information if they need more information for the implementation. So, and then for the world, I also offer this virtual course, which is organized by the European Bioinformatics Institute. It's called Cabana, so Capacity Building for Bioinformatics in Latin America. So I had the chance to teach last year, and this year we will repeat the training in Brazil. And of course, this is international. If you are interested, if you want to learn bioinformatics, please get in touch with Cabana folks. And who knows, we will see each other sooner or later, either in Brazil or virtually. So these are some people that I would like to thank a lot. These are all my supervisors in the course of the years. So Maria Eduarda, she is the president of the CRBio. It's the Brazilian Research Council in Biology. So this is Ana Benko, which is the president of the Brazilian Society of Genetics nowadays. So this is the Paulo Andrade, my professor of bioinformatics, genetics, Miguel Ortega, my PhD supervisor, and my 3 postdocs supervisors here, Reinhard Schneider, Miguel Andrade, and Mike Barnes. So I'm very thankful to them. So these are my colleagues at ITTM. It's a very young and successful company. Andreas is the CEO, Andreas Kremer, and Reinhard Schneider is the head of the board of directors. So we are very happy to collaborate with you either in a private or in a public-private partnership. So we are open for discussions, conversations, talks, and I thank you very much for your time. And I thank you a lot for being here until now. And I thank Tiago very much, Dr. Tiago Lopes, for this opportunity to have me here with you guys today. Thanks a lot. So I'm going to make Tiago host again. So Tiago, are you there? So, okay, please, Tiago.
Okay, thank you very much. This was indeed very, very interesting to see kind of the scale of this project, you know. This makes me think that, you know, the challenges are not only technical, but also kind of administrative, you know, to make this huge thing work, you know, and coordinate, you know, the data exchange and the kind of people involved and all the experiments and the analysis and so on. So I wonder if you could tell us a little bit about how was the administrative side? Like, was it like based in one country, centralized, or each group had their own staff? How did this thing work?
Yeah, you're right, Tiago. So for those international projects, what you have like is an organization in work packages, right? And you have normally one work package that's the project office. And the project office is the place where the project coordinators will be sitting and making sure that all the deliverables are written in time. So we have periodic reports that we need to fill those deliverables. And sometimes people like to write a deliverable in a way that you can use as a publication in the future. And then this is tracked very sensitively in the course of the weeks and the months of the project. We're talking about here a project of 5 years, 4 or 5 years. And then what you have is like yearly general assemblies where you report to the project office all of your results for the different work packages. And then there is a steering committee, which is made by independent researchers. So these independent researchers will evaluate whether or not the project is on a good course, right? And if not, they will pinpoint which places we should improve in order to make the project to achieve its goals. Okay. Right? So this is on the level of the whole project. So in the level of the individual work packages, you have 2 people that are responsible for. So normally one coming from the academia and the other one coming from the industry. So, and for example, in the moment I'm working for a project called TransBioLine that wants to discover what are the biomarkers of organ injury when you try a new medicament, a new drug. And then I need to do my work and coordinate the work of the other members of the work package. And in my case, I have to write the deliverable documents and send this to the project office.
Okay.
The project office will review and send to Brussels. And Brussels is the central spot where the Innovative Medicines Initiative, now they changed the name, it's called Innovative Health Initiatives. It's like where the coordination center for all the projects in Europe is sitting, and then they get in follow up with those deliverable documents.
I see.
Of course, it was a big challenge on the pandemic years,
Yeah.
and then we have like some delays here and there because it depends on the recruitment of patients. And if you don't have patients, of course, you don't have data. If you don't have data, you don't have results, and so forth.
Right. Yeah, this is something that I was also wondering about, how kind of was the impact.
Yeah.
So let's ask the audience, the folks who are attending online. So does anybody have any questions to ask to Dr. Adriano? Okay, so I got some here on the chat that folks sent me. It seems to be a more kind of technical question that I got here. So people are wondering about the hardware requirements to run this kind of large scale data analysis, you know, the things that you show with tranSMART, kind of how to store and how to process all these things. Are there like dozens of users or hundreds of users? How can you tell us a little bit about the hardware?
Yeah, that's a good question. And that's why I brought the slide about the research infrastructures. So the policy in Europe is that the European Commission wants every single scientist to be competitive per se. So it doesn't matter if you have like a really slow laptop in front of you. The important thing is that you can access via, SSH connection or secure connection, a center that provides you with more power. So, and more power is really like a tier 4 data center, let's put it like that. So in terms of data storage, you have institutes like EMBL, data library, where you can write your project and you can say, look, I'm interested to have access to this database, and I don't want to have a copy of the database on my laptop. So they will organize a kind of a partition so that you can have access under your user to that data that you requested remotely. So it's like you outsource this kind of a power for storage. In terms of data processing, it's the same idea. So you have like a huge processing centers there, normally at the European Bioinformatics Institute, in which you can write a project to use their research infrastructure. And your code should be written in a manner that is compatible with the analytical workflows that they use. In the case, they use Galaxy. And Galaxy is this popular data workflow platform. So they have a website called usegalaxy.org. And the idea is that you create the code on your home computer. So you export the code to GitHub. So you download the code from GitHub on your remote system, let's say, and then you run this code using the remote infrastructure. So at the end of the day, you have a really strong, powerful machine available for you within the limits of your project. Of course, you don't need a Ferrari to go to the bakery and buy bread. So the idea is that the resources will be evaluated by the IT team, and they will set up an environment that is, that's enough for you to run your analysis. So then these two aspects is already sorted, the data storage and the data processing.
I see. That's indeed very interesting.
So you just need to really apply for access, and normally, I think it's called European Open Source Cloud, EOSC, the name of the project.
I see. Very interesting. Very good to have all these resources available. So I have another technical question here that came in. Okay, so about the cancer project that you showed. I thought the same, that the normalization idea, that's a very clever one. That's a pretty good one, to normalize by the number of, times that it appeared, and then use that in conjunction with the kind of centrality of the protein. So the question here is, it seems that there are not all proteins present in this database. So it's kind of not the 20,000 proteins. So it seems that there is like a part missing or something like this. How did you deal with these missing links or this kind of the part that is still missing from this protein-protein interaction databases?
Yeah, so what we use is the consensus DB, right? We have this kind of, so the protein-protein mapping was made in another article. So I received already the table of the protein-protein interactions for this project. So I cannot tell you for sure how they handle that. Okay. So, but what you could do is like you could fetch, let's say, if you don't have the physical evidence of a protein interaction in a network, you could infer this by co-expression, for example. You could infer this by co-localization of proteins as well, or from text mining. So you could look at the literature using a text mining tool and see if proteins, they appear with a really high frequency in the literature. And you could infer that there is a biological interaction supported by text mining. But of course, this is not something that you have the physical evidence for, let's say, a yeast to hybrid system or a co-immunoprecipitation assay, something that the researcher went there in the lab, in the wet lab, and made the evidence that the proteins are connected to each other. A major database maintained by the Max Delbruck Institute for Molecular Genetics in Dahlem, Berlin, is the consensus, protein consensus DB. Okay. So the protein consensus DB shows a kind of a consensual map of protein interactions based on many different evidences. And this is what I would suggest you to look. So if you have a pair or a triplet or even a module of proteins interacting, and then you have your favorite protein that was left aside, you can try to connect your target protein to the module by using those databases. And then see if you could bring in new members to the interaction network. I don't know if I answered the question.
Yeah, I think so. And indeed, this is kind of very useful, you know, to understand kind of host response to infection, or what is a certain protein doing, like what are the interactions? And together with that normalization, it's a pretty good one. Mm-hmm. So I have a question of my own, but let me ask again, if anyone in audience, do you have any questions to ask Dr. Adriano, or you can also send me the text, don't worry, no problem.
I think there was NCCHD Mikami wanted to make a question. There was a hands up from them.
Okay, Mikami Sensei, do you have any questions? Well, I don't see anything.
No, nothing, thank you.
Okay.
Just realized you had your hands on, okay.
So, Okamura Sensei.
Hi, my name is Koji. Thank you for your presentation.
Koji, nice meeting you.
In your cancer classification study, you showed us a hierarchical clustering PCA analysis, and you performed artificial intelligence. But I couldn't really understand what feature for each gene, what feature did you use for the input? And I want to know the algorithm to identify candidate genes.
Yeah. Thank you. Thank you very much, Koji, for your question. You're welcome very much to ask here. The feature that we used were that calculation I showed you before. So the level of expression of one protein across the different samples of one cancer type. And then the example I showed was like the protein was overexpressed one time in 125 samples. And then that normalized value multiplied by the number of partners of the protein in the interaction network. So for each gene, we had this value, which is the normalized interactions count across all the samples. And then we compared what happened between the numbers. We compared the numbers of the highly aggressive and the low aggressive class by using these normalized counts. And that was the number that allowed us to distinguish the two different classes of cancer in high or low. And then for the next step, that was the classification of the sample, whether or not it's aggressive or not, was the presence and absence of the protein, because of course it's just one sample. And then instead of using 1 or 0 as a discrete value, we used the number of connections that the protein has. So if the protein, let's say, is present and it has 10 neighbors in the interaction network, we don't put 1, we put 10 there. And that's how we represent one sample. So presence or absence and the number of connections.
I see. So you used proteomics data, but not transcriptional data, right?
Both of them. The first information is that whether or not the gene is overexpressed in the sample. So if it's overexpressed or not, we mark 1, of course. And then we get the number of protein partners in the network, and then we just add to this one, we multiply 1 times 10. So then we have 2 evidences, the gene expression, yes or no, and then the number of partners in the network, the number of interactors, 10. So it's a very simple approach.
Okay, thank you. And my another question is, I don't know how to utilize your research result for clinical application.
Yeah, our idea is, first of all, we could get a sample of the tumor and verify in that sample whether or not the genes that we highlighted in our study, if they are overexpressed in the sample. So if they are overexpressed, like let's say most of the genes, let's say 7 out of 10 genes are overexpressed, then the idea is that that sample might be very aggressive to overall survival of the patient. And then you might go to the treatment that's the, let's say, the strongest treatment and the most effective for the tumor. On the other hand, if you have a tumor that is not so aggressive because it does not have the high levels of expression of our genes, you don't want to use the most aggressive treatment. You want to use a treatment that's a little bit more light, but works for that patient.
Okay.
So the idea then you will not cause any type of extra toxicity to the patient, because then you don't need such a strong drug to treat that specific patient once that you know the aggressiveness of the cancer is not that high.
Okay,
so this is the first application. And the second
Mhm.
application is that you can highlight the targets that you may study. You may study new drugs or new compounds that target those genes that we highlighted in this study.
Thank you.
You're welcome.
Great. So does anybody have any additional questions for Dr. Adriano? So I think that's also all. I mean, and just a minor comment for me, I like the concept of the education that you showed, so that's pretty nice, to have these education programs, these training programs to actually empower people to use these systems. I found this particularly very, very nice.
Yeah, I mean, this is something that I never moved away from. I used to be a bioinformatics associate professor in Brazil. And when I came to Europe since 2013, I've been more involved with research and hands-on activities on research. But, once a year at least, or even twice, like once a semester, I always try to offer those training courses and programs for summer, winter, autumn, spring training. And people that they want to start a new field in their career, I think it's a good opening doors opportunity, right? To get some new skills on your portfolio. And, it's always good to long life learning. It's something that I always motivate people to go forward.
Exactly. So, Dr. Adriano, I think We took a lot of time from your precious time. So I want to thank you again, for the opportunity to talk, to, exchange this idea, to tell us about these projects. They are all very interesting. And I hope we can collaborate soon.
Sure.
So with all that said, I thank you very much.
I thank you too, Dr. Lopez. So it is my pleasure really to talk to all the audience. As I said, it's really like a good opportunity to talk to, people from so far away from here. And I mean, I'm happy that we have this possibility. And I wish all the best for everyone and a lots of success for the global seminar sessions of your institute. Thanks a lot for the invitation.
All right. So thank you very much. Thank you very much, everyone, and goodbye.